my talk is going to be a little bit different from the ones that preceded me, and that a lot of what I'm going to show is more kind of things that I've done with the hope of illustrating ways that you can use Celomics. And so what I'm going to be talking about is largely divorced from the background of the science or the actual projects that were addressed, and more just kind of a how-to to give you a sense of that. So obviously we're talking about the CX7 instrument, the purpose of which has been made abundantly clear, is to simultaneously acquire image data and quantify it, and allowing you to do this, as Dr. Fu said, in a very high throughput manner where you can image many different wells, many different cells, and really get some robust statistical analysis. What I'm going to talk about today are two different bio applications that I use as my bread and butter for the lab, really for most analyses. And so some of these are not really what the bio application is designed for, but it seems to work the best for me and my knowledge base. And the one that I use the most, which you can't see here because of the zoom, the one I use the most is the colocalization application. And so my goal for talking about this is to provide an overview of the logic that you go through as you're designing these experiments and setting up the algorithm. I want to show you that this application is really broadly applicable, and it's not just for colocalization studies. And I'm going to try to illustrate some of these points, show some published and unpublished examples of us using this work. And the way that this basically works here is an illustration of a cell in this hypothetical experiment. We want to be able to quantify the number of these green dots in the cell. And I think from Dr. Fu's experiments, she did a lot of counting of dots and analysis of dots. And that's what I do a lot of as well. And so I was going to go through sort of the analysis in a step-by-step fashion. And for us, in most cases, we use a nuclear stain to help us identify the cell. And so our first step is in CX7 speak is to identify the primary object, which for us is synonymous with an individual cell, but does not have to be. This is just how we do things. Once we have identified the nucleus of the cell, typically we define the next step is to define your region of interest, which is the outline of the cell. And there are a couple of ways you could do this. Dr. Fu used cell mask, which is a reagent that will label all of the cell. For most, but not all of our applications, we can just tell the software to generate a ring or circle around the nucleus of a specified diameter. And that is usually good enough. And so you can see in this example here, this blue circle would be our ROI. It's not perfect. Sometimes it will extend outside of the boundary of the cell. Sometimes it won't cover all the cell. Sometimes we may miss some of the things that we want to measure. This is generally okay, using the caveat that these errors need to be, you need to be pretty sure that these errors are uniform across your sample. So if your treatments are changing the shape of the cells, or if you're plating different densities of cells, that can really impact this region of interest. And so that's stated here that, it's okay if you don't need the cell boundaries to align perfectly with your ROI. You can do this if the cell size or shape doesn't change between treatments or samples. Otherwise, a plasma membrane dye or the cell mask should really be used to more precisely label the region of interest. So the next step after you've defined your region of interest is to identify your targets. And in Celomics speaks, using this application, the target is what you actually want to measure. In this case, it's our dots. So you'll notice that all these dots, I've added two of them, they now have a black mask on them. And you can notice a few things. So in number 6 here, our object is star-shaped instead of round. In object 7, it's really big. And in object 8, it didn't do a great job of identifying that there were actually 2 dots there instead of 1. And in your first step, your object identification, you don't care. You want to cast a broad net, because in the next step, you're going to be throwing out the objects you don't like. And so you can really identify your objects based on size, shape, intensity of the neighboring pixels, etc. You can do segmentation, as I'll show you in a second, of objects by intensity or shape. And there's really a lot of geometry and math that goes into this. Fortunately, I think most of us get away with not knowing this, and we can use a lot of trial and error to really hone in on what exactly we want. And what we can do is we can visually compare what we're seeing on the masks to what our eyes would say. And so we can see, if you can see from the back of the room, there are actually 2 dots here in number 8, but only 1 mask. With a little bit of work, we can now segment those out to get 8 and 9. And again, at this stage, you're casting your net broadly. You want to get as many objects as you can, because in the next step, we're now doing our target verification. And here's where we will, through a variety of different category parameters, throw out what we think are not useful. For example, this star-shaped dot is not really what we want to measure. We don't know what it is, but we want to get rid of it. And so we can, based on shape and perimeter to area ratios and things of that nature. Same with this dot, maybe it's not real for whatever reason, we throw it out. I'll then skip over what we do next, but basically, once we've got all this set up, we can then move forward with our imaging and our now data acquisition in a way that is uniformly biased. I don't want to say it's unbiased, but it's uniformly biased. Okay, so what are some of the ways that I've used this co-localization bioapp in my work? One of the first things that we did, again, this is a lot of counting of dots, was to count the number of autophagosomes in cells that had been transfected with an siRNA library to knock down TREM proteins. TREM proteins there's a very large family of ubiquitin ligases that we hypothesized would be regulators of autophagy. And so initially we performed an siRNA screen of 68 human TREMs, and then assessed the abundance of autophagosomes because they're labeled with GFP fused to LC3B, which is an autophagosome marker, and then counted them by high content microscopy. The way our screen was set up, we could look for TREMs that, when knocked down, reduced the number of autophagosomes, which would be below this 3 standard deviation line here, and are similar to the untreated controls. We also could see ones that, when we knocked down the TREMs, we could see more autophagosomes, as typified by these that are above the line and are significantly higher than our treated controls. And in the end, what we did is, our N for these experiments is we did 3 plates per experiment, and for each well, there was 1 siRNA well per plate. We analyzed 500 cells. And as you can see, we got some very tight data that looked really nice. I'll show you some more data about TREM5 later, which we followed up on a lot. But so this is one application that absolutely could not have been done by confocal imaging and manual things alone. So in the last example, I showed you the quantification of dots in fixed cells. We've also gone a step further and looked at the quantitation of dots in live cells using the incubator that Dr. Desai talked about, where here our hypothesis was that TREM5, which I mentioned on the last slide, might contribute to mitophagy, that much we knew, by recruiting the mitophagy protein parkin to damage mitochondria. And so here what we're doing is we're quantitating the recruitment of YFP-labeled parkin to mitochondria in wild-type cells or cells in which we've knocked out TREM5. And we did this at different time points after mitochondrial uncoupling. And we got very nice data that showed that this hypothesis was not correct. But you can see the data and the responses in the Celomics over time. So this is another way that you can use this function by live microscopy. We've also used this co-localization app to quantify dots of representing protein-protein interactions. So this is using the proximity ligation assay, which measures proteins that are within 40 nanometers of each other, suggesting very close or even direct interactions. So the way this works is you have antibodies against your protein 1 and protein 2. Secondary antibodies that detect these are conjugated to oligos that, when they're close enough, they can form a circle. And then in a polymerase reaction, you can generate a lot more DNA that incorporates a fluorescent nucleotide. And then that gets read out as a red dot. And so here we're looking at TREM5, same protein, interacting with the ER-resident protein calnexin, because we had some data that suggested that mitochondrial damage induces the localization or the relocalization of TREM5 to ER mitochondrial contact sites. And so here, under untreated cells, you see very few of these red dots here in the image. And what you see, they're few, they're small, they're not many per cell, and they aren't very big. When we treat cells with a mitochondrial coupler, CCCP, now you can see visually there are a lot of these dots, and there are more of them, and they're bigger. And so here, each data point represents a single cell, and I believe we imaged 2,000 cells. So these assays, we've got one more. So all of these assays have been co-localization, but really only looking at one target. I want to show you one other use for this sort of assay. We had observed by confocal microscopy that when we damaged mitochondria, TREM5 relocalized from round dots, as you can see here, to, if you can appreciate, sort of these elongated, sometimes bent structures after mitochondrial damage, as you can see here. And so we wanted to develop an assay to measure that by high content microscopy. And so this was work done by Geneva Williams when she was a rotation student in the lab, I believe, where we could then separate these TREM5 structures based on their morphology. And the colors may be a little bit hard to appreciate, but here, see, if you see a magenta mask, it's identifying a round TREM5 structure. And then if you see a green mask, it's identifying a TREM5 structure that's more elongated. And we could, after treatment, we found that measuring the ratio between the long and the round, that's changed and increased with more long TREM5 structures after mitochondrial damage. And so this became a useful assay. We thought that we could use this to then identify the factors leading to this relocalization of TREM5 into these elongated structures at ER mitochondrial contact sites. Okay, so everything I've shown you now has been one target. Of course, the reason that the co-localization bioapp exists is actually to measure more than one target and actually measure co-localization, which of course you can do. And you can get measures of the quality of the co-localization. So this would be your Pearson's coefficient. So the number of 1 would be perfect co-localization. You can also measure the quantity of co-localization, which is often what we want to do and what I'm showing here. And you can do this for up to 3 color co-localization experiments. And so what we're showing here is TREM5 again. These TREM5 structures after mitochondrial damage with ivermectin or in untreated cells, and whether or not, and their abundance of co-localization with the autophagy protein ATG13. And so if you look at the masks, we have a red mask on the TREM5 structures, more of which are elongated here because we damaged the mitochondria. And then the purple mask is ATG13 that co-localizes with TREM5. And here we can see that ivermectin treatment increases that abundance although it didn't actually change the quality. The co-localization was just as good, but there was just more co-localization. Okay, so in the last second, I want to talk about the other bread and butter app that we use, and this is the spot detector application. And we don't actually use it much for spot detecting. We use it more for measuring the whole intensity of a cell. And I know that Solomics has a intensity measurement function. I don't like it as well. So what we're doing here is our cell again. Now we want to measure the total intensity of a protein within our region of interest, kind of like a single cell Western blot, where we're getting relative intensities. And so this would be an intensity profile along this hypothetical transect, where it goes through this green dots, we get a big peak. But you can see there's definitely some cytosolic localization of the protein. It's not just in peaks, but it's nuclear excluded. That's why it's down here. So what we do is we measure all of the fluorescence intensity above your background signal. And we've used this to measure, to do viral infectivity assays or nuclear translocation assays. And I'll show you just a couple examples real quickly here. Here we have cells that are expressing TRIM5 from rhesus macaques, which actually blocks infection by HIV, or human TRIM5, which does not. We're now infecting with different MOIs of HIV that expresses a green fluorescent protein. And so you can see that the rhesus TRIM5 cells, there's no green. In the negative control cells, the human TRIM5, there is green, and this is quantitated here. This is using this spot detector function, just measuring how much green is there in the cell. And then we also have found that this works really well for measuring nuclear localization of proteins. We've done NF-kappa B, we've done other transcription factors that are induced to go into the nucleus upon certain stimuli. And so here we're measuring, this was in collaboration with Kieran Boskar's lab, the abundance of NF-kappa B P65 within the nucleus after in control microglia or in microglia treated with pathogenic tau tangles. So happy to talk more about this, but this is basically how our lab, we've done many, many different assays, but if you know these two assays, you can pretty much do everything you want. And I will finish it there.